Hi, I'm Amy, and I will be talking about single nucleotide polymorphisms, or SNPs, variation in epiphytic genes compared across different Erwinia tasmaniensis strains. And so I'll first just talk about Erwinia tasmaniensis as a whole. It has been described as non-pathogenic epiphyte. Non-pathogenic meaning it doesn't cause disease. Epiphyte meaning that it can live on the surface of plants, but it's not parasitic. It doesn't do anything to the plant. It just lives there. And so currently there is only one strain that has been fully sequenced and published into like a full genome, and that is the strain ET199. Now, in my research and based on preliminary data that I have done, I believe that I have sequenced three other Erwinia tasmaniensis strains that have been isolated from nectar. Specifically, the species of the columbine flower, the picture here on the right is Aquilegia formosa. I have isolated these strains from this nectar specifically, and so I have named the three strains AK48, 49, and 50. It's just our naming system for these. But yeah, based on the data that we have, I believe these are three other strains of the same species of the Erwinia tasmaniensis. And so I'm also going to be talking about or comparing in my project Erwinia amylivora, which is the type species for the Erwinia genus. And this just means that it sort of serves as like the reference. This is sort of like the reference for my reference for strain ET199. And this bacteria is pathogenic. And so it does cause disease. It causes the fire blight disease in plants, and it's very, very destructive. It can knock out like a whole orchard in one season only. So it's very destructive. And so this also serves as like a reference for my project. And so then the motivation for this project was I wanted to do something with Erwinia tasmaniensis, since that's kind of my my thesis is on this bacteria. And so again, it is epiphytic and it is non-pathogenic as well. And so some interesting properties of epiphytic survival is surface adhesion. So how well can it stick to a surface and, not get blown off by the wind or animals coming and rubbing on the leaves and knocking it off. And also osmotic tolerance, antimicrobial resistance, biofilm formation. So how well can it form thick biofilms in it, and how well can it also serves as like a method for sticking, how well can it stick, and then also quorum sensing. And so continuing with the motivation, there are a few genes that are related to some of those properties. And so these are some of them that I found the most interesting. And so there's a gene called OTS A and B, which is a trehalose, I hope I'm saying that right, biosynthesis, where the trehalose forms a sort of glassy matrix that maintains the structural integrity and even like stabilizes proteins and membranes when it is dehydrated. So when no water is available, it still protects important cellular components, and so therefore it can survive in those conditions. And then there is a gene called LSCA, which produces levain sucrase, which is an enzyme that converts sucrose to levain. And so levain is also extremely important in biofilm formation. And so that's sort of the material or the product, I guess is a better word for it, that produces the sort of viscosity and almost like stickiness of the biofilm. So it's kind of what helps makes it stick and keeps everything in place from movement. And so then the other gene sort of sequence was FIMABCD, which produces a type 1 fimbriae, which again produces hair-like appendages on the bacterial surface. So not to be confused with flagella, but this produces sort of like the fuzzy look of bacteria that sometimes you can see, or like with this picture up on the top right, sort of that like fuzzy hair-like surface. And so with all of that together, the question that I'm asking is, are genes related to epiphytic behavior, osmotic stress, biofilm formation, and adhesion present, and are they polymorphic between the Erwinia tasmaniensis strain ET199, my Erwinia tasmaniensis strains AQ48, 49, and 50, and also Erwinia amylivora? And if these genes are present, how many SNPs separate each gene across the strains or species? And so, from what I found, no studies have compared intraspecies genomic variation in these genes for the Erwinia genus. And also, another point is we do not know whether uh, epiphytic adaptations are conserved, or are they strain specific? Are they sort of like adapted once they're established? So, I also found that kind of interesting. And so, my approach going into this was, again, the Erwinia tasmaniensis strain already has a genome, so I could easily get that from NCBI. And then my Erwinia tasmaniensis strains, I've already assembled and annotated them. I've done that in KBase, so already have that established. And then the Erwinia amylivora, again, it's already up there in NCBI, so super easy, just download that. And then the genes of interest, again, the OTS A and B, LS C A, and FIM ABCD. That's what I'm going to be focusing on. And so, again, using the AWS servers and terminals where I plan to do the majority of this. And so I found a tool called Panaroo, which is used for pangenome analysis. So, from what I've read, it essentially checks for whether certain genes are present or absent in a bunch of different genomes. And that's exactly what I'm looking for. So, that's the tool that I decided to use, and it's what it sounds good. And then for SNP calling, again, like we've used in class, SAM tools and BCFT tools. And then I plan to make a summary table of whether genes are present or absent and the SNP count. I could do this using like Python in the terminal, but I think maybe R would be a better approach for this. I don't know. We'll see when I get there what seems easier at the time, but that's my plan. And then some possible results. I could see that, maybe the genes are conserved, so very few SNPs are present, which could mean that the epiphytic traits are maintained across the strains or the species. And then I could also see variation within those genes, which could indicate, certain adaptations to different plant habitats, maybe. And also, a possible result could be gene absence in some strains, which means that maybe these are accepted through horizontal gene transfer. And then we have a better understanding of how these genes come to be and how their behavior comes to be. And so, with that, these are the references that I used, and that concludes my presentation.